Okay, so today we are going to cover the last part of types of adaptive immunity. So last week we learned about the cell-mediated immunity, different types of cell components that involve, and its mechanism of action, right? So I received few questions from some of you who reached me personally asking about further explanation on previous topics, and I'm happy that you are currently doing revision of what we already learned. Okay, so this week actually will be my last class with all of you, and next week we are going to have our first quiz, which account for 25% for your 100% of assessment. So today we are going to cover the humoral immunity. Okay, so at the end of this lecture, I expect you to be able to describe the components of humoral immunity and explain the relationship between different cells of humoral immunity, which is the B lymphocyte, plasma cells, and memory cells. And also you should be able to describe the major steps involved in the activation, proliferation, and differentiation of B cells in antibody-mediated immunity, and also differentiate the different classes of antibody, and also how actually antibody works. Okay, so this is your lecture objective for today. So what we are going to cover today is the B lymphocyte cells activation, proliferation and differentiation of B cells, antigen recognition by B cells, antibody basic structure, antibody-dependent cell-mediated cytotoxicity, and classification of antibody. Okay, before I proceed with the introduction part, let me make it clear. So the other class in the morning just now, they are having, I understand your poor connectivity or unstable connection at in campus. Because I'm also having same situation. My network is always unstable, even at office. So let me know if you having problem or there's some lagging during my lecture. Just let me know, and I will try to find other suitable place or better place. Okay, and let me know if you like to share device, like you cannot cope with your poor connectivity, then I will allow you, uh, you can sharing device, okay, with your friends. Okay, just let me know if anything, because the other class just now, there's like almost like 30 minutes some internet problem, and they inform me late. So please bear with it. Just let me know if you're having problem with that. Okay, all right. So the overview of immune system. So we already covered the innate immunity. Okay, everyone clear about innate immunity and adaptive immunity, divided by T lymphocyte cell-mediated immunity and B lymphocyte humoral immunity. So last week, just want to recall slightly about last week, what we learned last week. So cell-mediated immunity mainly involve the T lymphocyte cells, and initiation of the T lymphocyte will initiate further immunological response, right? But for today, humoral immunity, the main cells that will involve is the B lymphocytes, that later on will initiate the differentiation into the plasma cell that are able to synthesize and release the antibodies. Okay, the humoral immunity, which is a third line of defense, okay, it is mediated by antibodies which are produced by activated B cells. And it usually involve in immune response to encounter particularly microbes and toxins that are able to, penetrate our body, already past the first and the second line defense. Okay, and the antibody may bind to the microbes to promote their phagocytosis in order to eliminate the function. So the antibody will first recognize the microbial antigen, then it will neutralize the infectivity and target the microbe for degradation. That is the mechanism of action of antibody-mediated immunity. Okay. So the B cells that involve in humoral immunity is produce and mature in bone marrow. So the B refer to the bone marrow. And you have to know the specific site which they are accumulate, which is at the germinal center, which include the white pulp of the spleen and the cortex of the lymph node. And it is estimated about 10 million different clones of B cells available, which makes different types of antibodies. And humoral immunity involves the activation of beta cells into plasma cells, and plasma cells have the ability to produce and secrete the antibodies that will be released to blood and lymphatic fluid to exert its immune response. Okay, a structure of beta cells, the main structure very important that you need to know is the not beta cells, but B cells is B cells receptor or BCR. So this is the structure of BCR. It is antigen specific receptor and it has two identical heavy chain in purple here and connected with disulfide bond with the light chain in green. Okay, and it makes it in a form of Y shape. Okay, there are two antigen binding sites exposed to the exterior of the B cells that are involved in binding to specific pathogen epitopes to initiate the activation process. So if you remember, last week I explained about the T helper cells activation, that the T cell receptor only interact with antigen epitopes that presented the MHC1 and MHC2 on its surface. Okay, but for BCR, it do not require antigen presentation with MHC. They can directly interact with epitopes on free antigens or with epitopes displayed on the surface of intact pathogens. And TCR also only recognize protein epitopes that already the pathogen digested by the lysozyme express the peptide antigen on the surface of the APC, right? It only recognize the protein epitopes, the peptide antigen. But for the BCR, it can recognize epitope associated with different molecular classes, not only protein, but for example, the polysaccharides, lipopolysaccharides. Okay. So for everyone clear with the B cell receptor, so we need to know this first in order to learn how the B cells is activated. Okay. So the activation of B cells can be divided into two different pathways. The first one is T cell independent activation of B cells and T cell dependent activation of B cells. T cell independent, which is do not require the help from T helper cells. So it can be a direct binding to polysaccharide, lipopolysaccharide, or other non-protein antigens without antigen processing and presentation to the T helper. But for the second pathway, the cell-dependent activation of B cells, it requires B cell to function as APC, presenting the protein epitope with MHC2 to helper T cells. So basically, the T cell dependent will involve the action of the helper T cells. The T cell independent without the T cells help. Okay. So I put an illustration here. So you do not know how, you do not have to know like very comprehensive on each of the steps that leads to the secretion of the antibodies. So just you know that the difference between these two pathways and is already enough. Okay, for the T cell dependent, so it depends on the activated T cells for the activation of the B cells. That later the B cells will initiate its proliferation and differentiation into plasma cell to release the antibodies. Okay, but for T cell independent pathway, it involves direct binding to the peptide to the antigen, and then also requires second signal. For example, interaction with other receptor, a toll-like receptor, for example, and then initiate the plasma cells to release the antibodies. So this is the difference with the T cells and without the T cells. Okay, let's look at the first one, the T cell independent activation of B cells. So the first signal for activation is the repetitive epitopes on the antigen peptide form a cross linkage with the multiple B cells receptor. here. So this is a naive mature B cells with the BCRs on its surface, and then the repetitive epitopes on the T-independent antigen. It could be peptide, liposaccharide, attached or making complex cross-linkage with the BCR. And then it require second signal for activation, which is interaction with toll-like receptor or with other factor, for example, the complement system. Only then this beta cell will be activated, and then it will undergo clonal proliferation and differentiate into a plasma cells. So plasma cells is actually the B cells, but it's in differentiated form. Okay, it's already loose. It's BCR at the surface, but it has a special abilities to secrete, to produce and secrete the antibodies. So that is the plasma cell. And the types of antibody that released by the plasma cells upon activation of the B cells is the pentameric IgM. So there are 5 classes of pentameric of the antibody, and the first antibody to be secreted, you have to know that it is pentameric IgM. We will learn about the different classes of antibody later on. Okay, all right. So most important thing you need to know that the T cell independent is short-lived and does not result in the production of memory B cells. The memory B cells is required to initiate the subsequent attack of the same antigen in our body. Okay, so in this pathway, there is no memory B cell is produced. Okay, you have to be clear with that. Okay, so let's see. So this illustration I illustrated to represent the B cells with the BCR, the receptor on its surface, which recognize the repetitive epitopes on the pathogenic bacterial cells. Okay, so upon making contact, the beta cell will be activated. That it will initiate the proliferation and differentiation into plasma cells to produce and secrete the IgM, the pentameric IgM antibody. Okay. So this is a much more simple illustration. Okay, that is without the involvement of the T helper cells. And the T cell dependent activation of B cells require the help from the T cells. So how? But first, you have to know that the T cell dependent, it can occur in response to free protein antigen or to protein antigen associated with an intact pathogen that express the antigens on its surface. Either one, both will result in internalization of the antigen. Internalization means penetrating of the peptide into the B cells. Okay. Once internalized inside the B cells, the protein antigen is processed and presented with MHC class 2 on its surface. So who recognize the MHC class 2? It's the T helper, right? So here, upon recognition with the antigen epitopes on the bacterium, so the peptide will be internalized into the cells and then making complex with the MHC class 2. That this complex will be recognized by the CD4 of the T helper cells, right? If you still remember. Okay, and the coordination between these B and T cells, we call it as a linkage recognition. Okay. What happened after that is that the B cells is activated. Once B cell is activated, the T helper type 2 will produce and secrete the cytokines that activated the B cell and cause proliferation into clonal daughter or plasma cells. Yeah. So upon making contact, the cytokine will be released. And if you remember, cytokine involved in autocrine mechanism, right? Which initiate the B cell to proliferate themselves. Okay, and proliferate to become a plasma cell, a clonal daughter cells. Number one. And also the release of cytokines also will initiate the B cells by time to form or produce the memory B cells. The memory B cells will be stored in a body that will be active upon the subsequent attack by the same antigen on bacterium later on. Okay, so this is the memory B cells. So in the T cell independent, there is no production of the memory B cells. But in T cell dependence, you have to know plasma cells also being differentiated and produced, and as well as the memory B cells. And the plasma cells that have the ability to produce and secrete antibody will release the pentameric IgM, the first antibody types of antibody that being produced. Okay. Can you clear differentiation between the cell T cell independent and T cell dependent? Is everyone clear? Before I proceed, all right?
Okay, Doctor.
Okay, Doctor.
Right. All right. So as I said, there are 5 types of different classes of antibodies. The first antibody produced is pentameric IgM. So what about the others? So upon initial secretion of the IgM, the pentameric IgM, cytokines secreted by the Th2 will stimulate the other plasma cell to switch from producing the IgM to produce other antibodies, for example, IgG, IgA, IgE. Okay, so this process is called class switching or isotype switching by the plasma cells. Okay, so the other class of antibody is also needed to initiate the immune response to encounter the pathogen. Okay, so you have to know about the class switching or isotype switching, how different types of antibody is being produced by the plasma cell. Okay. Okay, so what is the difference between plasma cells and memory cells? So the plasma cell, when activated B cells will multiply rapidly by cell division and then clone itself to produce a population of identical cells, which is the plasma cell. And plasma is actually a differentiated form of the B cells. The B cells have B cell receptor on its surface, but on differentiated plasma cells, the B cell receptor is abolished, diminished. There is no more B cell receptor on the plasma cells. But plasma cells have this special ability to produce and secrete the antibodies. But B cells itself cannot secrete the antibodies. It need to be activated first and differentiated into the plasma cells. So that is a plasma cell. And memory B cells, some activated B cells also will remain in the body as a memory cells upon differentiation into its clonal daughter. Okay, that the B cells, the memory B cell will be used for the subsequent attack. Okay, so that when a person encounter an attack to from the same peptide antigen, the pathogen infection infects again the body, that it will be destroyed by the memory B cell that already being produced during the first attack. So before any symptoms show, the pathogens already eliminated by the memory B cells. So that is different between plasma and memory B cells. Okay, so I put the complete illustration here. So this is the antigen presenting cell with MHC antigens complex on its surface, recognized by the T helper cells and activated T helper cells. We further recognize or cross-link with the B cell expressing the MHC class 2 with peptide antigens on its surface. And then upon activation, it will be proliferate and differentiate into plasma cells that initiate by the cytokine stimulation. that the plasma cell will release the pentameric IgM, and later on class switching or isotype switching will occur, that the plasma cell will initiate different types, production of different types of other antibodies. Okay. So we finish with the B cells activation. Now let's look at what is the types of antibody. So antibodies is a specific protein produced by plasma cells, the B lymphocyte producing antibody, in response to antigen, and it is a member of the class of immunoglobulins. And when their body encounters a pathogen for the first time, the B cell produce antibody that specifically recognize antigen associated with that particular pathogen. So that is why we call the adaptive immunity is specific, because one specific antibody recognize a specific antigen of a particular pathogen. So this is example one type of antigens with its epitopes expressed on the surface, and that the antibody will recognize a specific epitopes of the antigen, and it will form lock and key to form an antigen antibody complexes. So this is the basic structure of um, antibody. So it consists of four proteins chain held together by the sulfide bond. So it have the heavy chain and light chain here in a form of a Y-shaped structure. So you have to know there are two functional components or functional region of the antibodies. The Fab region here, here, and also the FC region. So the Fab region can be divided into variable region, where the antigen binding occur, and also the constant region. Okay, so this is the depends on, it will change depends on different types of the antigens, epitopes on the antigens. Okay, so it gives the specificity of the antibody to bind to specific antigen, and the constant region usually determines the mechanism used to destroy the antigens upon the antigen antibody complexes. So this part will determine what type of mechanism that will be used to destroy the antigens. Okay. And for FC regions, this is a fragment crystallizable part that activate the immune system, and also it is the site of complement factor binding um, to be presented to the phagocytic cells during the antibody-mediated opsonization. Okay. So this is all five kinds of antibodies: IgD, IgE, IgG, IgA, IgM. All of them are actually different in structure if you look at closely. Okay. So let's learn by one by one. So IgG, the highest percentage in plasma, 70 to 75 of total immunoglobulin, and it is secreted all body fluids in high quantity upon secondary exposure. And it is in a form of monomer with two Fab site, 1 and 2. And it functions to neutralize microbes and toxin, opsonize antigen for phagocytosis, and activate the complement system. And the special abilities, because it is the smallest, it can penetrate efficiently into tissue spaces, and is the only antibody class with the ability to cross the placenta and protecting the developing fetus. Okay, so this is the IgG. So what about IgM? So this is a pentameric IgM. So it is pentamer with 10 Fab site, 2, 4, 6, 8, and 10. Okay, it is called pentamer with 10 Fab site. So it account for 5 to 10% of plasma, and secreted through the body, blood, and lymph, and secreted initially during primary infection. So it functions to neutralize, agglutinate, and complement activation, and the monomer form serve as a B cells receptor. So the first antibody produced and secreted by the B cells during the primary and secondary immune response, and it is the biggest because it is in pentamer structure with high avidity to combine strength of all binding site, because it has 10 Fab site that have a strong ability to bind to the antigen. Okay. So how about IgA? So it is account for 10 to 15% of plasma, and it is a body secretion, and it is in a form of dimer. One monomer. It is a dimer with 4 Fab sites. So please remember the structure, so that when I ask you, when I give you one picture of antibody, then you will be able to recognize it easily. Okay, so it function in neutralization and trapping of pathogens in mucus, and special abilities to protect body surfaces that are exposed to outside foreign substance. IgA from breast milk will protect the newborn against infection. And how about IgD? It is account for 0.25%. It present on the surface of B lymphocyte and in a form of monomer with 2 Fab site, and is a membrane-bound monomer found on surface of B cells, where it serves as an antigen receptor. So it acts as a B lymphocyte activation signal and play role in allergic reaction. Okay. So the last one is IgE, 0.05% of plasma, secreted in lung, skin, mucous membrane, on mast cells, and basophil. And it is also in a form of monomer with 2 Fab sites. And it involved in activation of basophil and mast cells against parasite and allergens, and stimulate mast cell and basophil to release potent pro-inflammatory mediators, and also stimulate allergic reaction. So that is this one may be mistake. So I will correct this one. It's actually IgE. This one, IgE. Okay. So I already deposited the lecture note, but please, can you make it changes by yourself? Okay.
Alright.
All right. Okay. Now, how antibodies work? So it works in 3 distinct ways: preventing pathogens from entering or damaging cell by neutralization and agglutination, stimulating removal of pathogens by macrophages and other cells by opsonization or antibody-dependent cell-mediated, and also trigger destruction of the pathogens by activating the complement system. Okay, so for neutralization, so the antibody will bind to the epitopes on the surface of the pathogens or the toxin and prevent their attachment to the cells. So every pathogens or toxin, for example, has epitopes expressing on its surface that will making contact to enter the host cells. So what antibody do is that antibody will bind and masking or hinder the epitopes from being exposed or interact with the host cells to prevent their attachment to the cells. So making the viruses or the pathogens less potent. Okay, so that is neutralization. And opsonization. So we learn opsonization, the coating of the complementary on the surface of the pathogen. So same process happens for the antibody, where the antibody will coating the surface of the pathogens, binding to the epitopes on the surface of the pathogen, and assist the phagocyte to bind and facilitate the phagocytosis. For example, the macrophage here, we recognize the FC regions of the antibodies, and then we'll facilitate upon recognition, we facilitate the macrophage to initiate the phagocytosis. Okay, that is opsonization, coating of the antibody on the surface of the pathogens. And agglutinating and precipitating antigens. So some bacteria can be excluded from the body by causing agglutination or clumping together to make them less likely to spread. Okay, so it involves a cross-linking of pathogens by antibody to create large aggregates. And main antibody that involved in agglutination is IgM antibody with uh, pentameric IgM with 10 Fab site. So all these antigen binding site will recognize the epitopes on the bacteria and making them to form a clump or agglutination. That is the function of um, the agglutination. Okay, so when the bacteria agglutinate, clumping together, it make them um, less likely to spread. Okay. Okay. And activating the complement system also will be activated by the antibody to promote the inflammatory response and recruit the phagocytes to the site of infection, enhancing phagocytosis and killing gram-negative bacteria with a membrane attack complex, the MAX. Okay. So we learned about the mechanism of complement protein before, right? So it is the same. So the last one, antibody-dependent cell-mediated cytotoxicity, ADCC. So usually to enhance the killing of larger pathogens that cannot be phagocytosed by a normal way. Okay, so it needs the antibody to help to encounter this bigger pathogen. So what antibody do is that the Fab region of IgG antibody will bind to the large pathogens. So this is the Fab regions of the IgG antibody will bind to the epitopes of the antigens of a large pathogens, and the Fc region on the effector cell, for example, the natural killer cells that initiate this ADCC, will bind to the Fc regions of the antibody here and making close contact and interaction of the natural killer cells and the pathogens. Okay, so this is the pathogens with the epitope exposed and interact with the Fab region of the antibody, and then the antibody also have the Fc region exposed here that will be recognized by the Fc receptor on the natural killer cells. And upon close proximity or close contact of the NK cells and the antibody, it will initiate the NK cell to release the cytotoxins, for example, the porphyrin or the granzyme on the surface of the pathogens that later on will create a pore that induce the cell to enter the cell death program or apoptosis and eventually kill the pathogens. That is the antibody-dependent cell-mediated cytotoxicity. Okay, so this is the last slide on how the antibody works. Okay, so with that we finish the lecture for today, and I open for any question that you want to ask. You can ask me right now.